Hello, and welcome back. We're going to now discuss a little bit about eukaryotic cell division. All right. With eukaryotic cell division, we fall into one of two categories. All right. We've got mitosis, which is going to be a type of asexual reproduction. So the cells that come from mitosis are going to be genetically identical. All right. We've got meiosis. This is going to be generate the cells that are used in sexual reproduction. And these are going to be genetically diverse. Okay? So not all of the cells are going to be the same. Now, in terms of the cells of the body that undergo meiosis, there are going to be two types of cells that undergo meiosis. All right? You've got the egg in females. You've got the sperm in males. Those are the only two cells in the human body that will undergo meiosis. Okay? So if you're female, the one cell type that you've got that undergoes meiosis are the egg cells. If you're male, the one cell type you've got in your body that undergoes meiosis is the sperm cell. Okay? The cells that undergo mitosis are going to be everything else. So the cells of your kidney, your liver, your skin, your lung, all of those are going to give rise to new cells by the process of mitosis. All right? And when we talk about mitosis, I'm going to talk about specifically the stages of mitosis in the next videos. But before we get to the stages of mitosis, I think what we have to talk about first is how the DNA is going to be organized. Because eukaryotic cells have much more DNA than a prokaryotic cell does. So cell division is going to get a lot more complicated for eukaryotic cells versus prokaryotic cells. Okay? And the DNA is going to be organized in the eukaryotic cells into chromosomes. Okay? Those chromosomes are going to consist of DNA and protein. Okay? So we've got, I'm going to use the squiggly line to represent our DNA here. Okay? What's going to happen? That DNA is very thin and very fragile. Okay? So when that cell undergoes cell division, it wants to make sure that the DNA doesn't break and that it doesn't lose anything. So what's got to happen is that DNA essentially has to be packed up. Much like you would pack up your house to move, the DNA is going to kind of pack up itself so that it can safely be allocated into each cell. All right? And what's going to happen kind of at that first level is that DNA is going to kind of coil around itself, well, around a protein first. And that protein it's going to coil around are known as histones. Okay? Then what's going to happen, you're going to get more coiling that's going to kind of happen. And those histones and DNA will kind of coil around each other. Okay? The blue dots I'm drawing here, again, these are going to be histones. These are not cells. Okay, I know it can look similar. These are going to coil up, and eventually what's going to happen, all right, is that DNA and the histone will coil up into a nice tight structure. And I'll draw the histones in here like that. Okay. And then we're going to have a little bit that connects those two. Okay. What that's going to do, it's going to coil up into our chromosome. All right? And with human chromosomes, the way things are going to organize, there's going to be a P arm and a Q arm associated with the chromosome. Now, in general, the P arm tends to be a little bit longer than the Q arm, but in some of the chromosomes that I'll put up for you to look at, they look pretty close, okay? But that's going to be a way that the human chromosomes are going to organize. There's going to be a little bit of protein that kind of hangs in and holds those together. This pink bit is going to come into play when we talk about how the chromosomes are allocated or separated into the two new cells. And I'll talk more about that structure then, okay? But for now, I just want to show you what the chromosome is. So usually the way I draw chromosomes, I'm not going to go quite that complicated. I draw them a bit like this, okay? So this would be a single chromosome. When a chromosome undergoes DNA replication, because we're going to have that DNA replicated before we can start cell division. What'll happen, and how I'll kind of draw those, it's going to look more like an X, okay? So this is going to be a chromosome that has copied its DNA, all right? So that's what you're going to look for. The X's mean it's undergone DNA replication, all right? And what we refer those to, all right, because that's going to have a special name, those are sister chromatids, okay? With the individual chromosome being referred to as a chromatid, okay? And I guess I should define this. I don't know why I would wait, but what holds those together are the centromere, okay? So there's a centromere here, there's a centromere here. Those are going to hold the P and the Q arm together. The centromere is also going to be where you're going to hold the sister chromatids together as well, okay? When we undergo cell division, when we undergo mitosis, what'll happen, those sister chromatids will separate. And what's going to happen, you're going to have one chromatid go to one daughter cell, one chromatid go to the other daughter cell, okay? Because we want to divvy up the information equally between the two cells, because those chromosomes represent the instruction manual for the cell, okay? So those are our sister chromatids. What I'll do next, I'm going to talk a little bit about the stages. And I'm going to go into some more detail on this in the next video, okay? When we're undergoing the process of cell replication, this is very often terms called the cell cycle, because it's going to repeat itself as needed for that particular cell, okay? And in terms of the cell cycle, we're going to have a couple different stages. We're going to have interphase. This is kind of, some books kind of describe it as a resting stage or a quiescent stage, quiescent meaning it's not actively undergoing cell division, okay? But that interphase stage can vary a lot for different cells, okay? Then we've got interphase, which is going to be separated into a couple of steps. We've got G1, G2, oops, get ahead of myself. Let me get rid of that. Because before we go to G1,